Okay, so before we start going over the slides really quickly, we just want to make it very clear. This is not going to count for any DES credits or AEP or anything like that. We're doing this as a way to just kind of help you guys out for your exams and review the past week's material, and also to help ourselves out because we're reviewing for our BSCE. But we also just wanted to let you guys know before you get into this that this doesn't count for your requirements for AEP.
Yes. Good reminder, Sarah. Okay, we're going to kind of go in order of the way you guys had the lectures this week. I know it was kind of a dense week, and I think the next week's going to be the same, but just hang in there because after FTM, it's going to get like a lot better, I promise. But we'll start with the population genetics. Okay, so I know there was a lot of material on this. I know there was a lot of DLAs. I know that it's kind of tedious. There's math involved. Before we start talking about Hardy-Weinberg, I would definitely recommend going off of the exam breakdown thing we were just talking about. If you don't have the foundations of the genetics down super well, make sure you go back over that and become comfortable with that, because with Hardy-Weinberg and with the population genetic stuff, you need to know like the back of your hand if something is an autosomal recessive or dominant or X-linked, because that's going to affect the rest of the math you do. So if you don't have that foundation down super well, this is going to be a little bit harder. And there is math involved, so you don't want to be wasting time on your exam thinking about things and equations and things like that. So make sure you have the foundation on this stuff. But know for sure that you have to have these assumptions. They have to be met in order for the Hardy-Weinberg equilibrium to even be applicable. So you have to have that large population with the random mating, no new mutations and no migration, things like that. I would definitely make sure you know these factors that can cause the genetic variation, because as always, it's an exception. So exceptions are very testable. And I know we for sure had questions on founder effect and new mutation, genetic drift, things like that. And I'll get a little bit more into those after we go over the basic stuff. This is a screenshot from first aid. We love first aid. It's great. So this is just reiterating the stuff that Dr. Sobering gave you. This is just saying, your P and your Q, they're the alleles. P is going to be the normal allele, and your Q is going to be the disease-causing allele. So we can actually skip past this one, but that's good for y'all to review. Okay, so like I said, starting with the basics, just make sure you kind of understand this concept. It makes a lot of sense if you just think about it. You just have to remember that everybody has two alleles. So the homozygous for the normal allele, there's going to be two copies of that, and the heterozygote is going to have one copy of each. And then the homozygous for the recessive or the disease-causing allele is going to have two of those affected copies. So make sure that you're aware that instead of making your total out of 100, hypothetically for the 100%, there are going to be 200 total alleles. And it gets a little bit trickier thinking about the heterozygotes, in my opinion, because you need to count one of the P's and one of the Q's, or here denoted as the M and the N. Okay, so here's the equation. I'm sure you guys have had a lot of practice with this week, hopefully. But so Dr. Sobering likes to, this is taught a couple of different ways. There's some textbooks out there that like to say your P is going to be your dominant allele and your Q is going to be recessive, whatever. Just the way he likes to teach it and the way he told us to is let P be the functional allele, and then Q is going to be the disease-causing allele. And as long as you keep that consistent, you'll be okay in these. And I actually think that's explained on the next slide better. Yeah, so just make sure you keep it consistent. This is one of those equations that really, if you switched everything up, it would be okay. You just need to be consistent about it. But the way he teaches it, in my opinion, is the clearest and the easiest way to understand it. So your P is going to be the functional allele, which is always going to be most commonly found. And then the Q will be the disease-causing allele. Okay, just again, reiterating it. So if someone's homozygous normal with that, the big A, you're gonna have, that's gonna be the P. So that means that the P squared is representing those two normal alleles. And the 2PQ is going to be representing the heterozygotes because they're going to have one of each. And then the Q squared is going to be the homozygous recessive person for the disease-causing allele. So that will always be what we're actually going to be focusing on for the most part, is the Q, because in a lot of these we can simplify out the P, which I think is next on here. Okay, so with these, you have to make sure that you know the inheritance pattern of the disease in question, because they are no longer going to be, well, they didn't do that in the first place, but before on your first exam, it was more like you kind of needed to figure out if it was autosomal dominant or recessive. Now you're going to be building on that. So you're going to have the same types of questions, but instead of needing to know that, you just need to know that already. Like they'll give you cystic fibrosis and you need to know that that's autosomal recessive, and then be able to apply that in the Hardy-Weinberg equations, because you're going to use your Q in reference to your incidence, but they're different for each of the inheritance patterns. So you need to know those. So for autosomal recessive, which I would argue is probably one of the most common one used. Yeah, they use that, like, because there's a bunch of those inheritances that are autosomal recessive, but Q squared is going to be the same thing as the incidence. So the incidence in the population is going to be the number of affected people. Q squared is going to be the incidence, and that's because in order to be affected with the autosomal recessive disorder, you have to have both of those alleles. So that's why it's going to be Q squared. You have to have both of the disease-causing alleles. You'll need to know how to interchange these a lot. So knowing that Q squared is going to equal I, and that Q is the square root of I, make sure you don't mix those up, because it can get a little bit tricky, especially with the fractions. So just keep that in mind. I just memorized these. It made it a lot easier. Yeah, this is kind of just building on that. So Q squared, the proportion of people that have it. So this is going to be, the Q squared is the incidence. So if you're trying to solve for just Q, for example, if you're getting asked something where you need to find out the carriers or something further, you'll need to solve for Q. So you just need to remember that if Q squared is the incidence, then you're going to need to take the square root of the incidence to find the Q. And then to find the carriers, that's when you have that 2PQ. And in AR, that's because they're going to be heterozygotes if they're a carrier for the disease-causing allele. So that's why that's that 2PQ. But it makes it a little bit easier that P is always going to be very close to 1. So you can actually leave out the P in these equations, which is pretty awesome. It makes it very simple. So leave out the P and you're going to use 2Q as the incidence, which makes Q half of the incidence. So just remember, you need to be able to interchange those, knowing that the incidence is 2Q, or if you're needing to solve for Q, then Q is going to be 1/2 times the incidence. Yeah, and just building off of that, I think for every inheritance pattern that you cover in this course, at least, the P can be simplified to just the Q, because all of the inheritance patterns that you guys do, the P would have to be like a homozygote for the disease, for the one that has a very, very, very small possibility of happening. So you can simplify that out and take the P out of the 2PQ, and you can almost always just use the 2Q, because yeah, that less than 5% thing. And I know Sebring went over that really well with you guys. Autosomal dominant, the incidence is going to be now that 2PQ, because if you remember, autosomal dominant disorders manifest themselves in the heterozygous state. So that's why if you think of that middle equation, the 2PQ, as the heterozygotes, that's going to be how many people are affected, or the incidence with the disorder. So that's why your 2PQ is going to be the incidence, but you can also take out the P as well. So it's just going to be 2Q equals the incidence, or Q is half of the incidence.
Sorry, I have a question.
Okay.
For the carrier frequency of the autosomal recessive, the equation you had as Q is equal to half times incident. In that situation, what are we essentially looking at? are we trying to find what the carrier frequency is, or are we trying to find what the allele frequency is if we're given the incidence of the disease? Like,
if you're given the incidence and you're looking for the carriers, you've got to remember that you're going to have more carriers than you are going to have affected people. So you're going to take your incidence and it's going to be twice that, because if someone's affected, they have 2 copies of the disease causing allele. If somebody is not affected, they're carrying the allele. And that's why they say, you know, there's a lot of autosomal recessive carriers out there that don't even know it because they're not going to show symptoms there's a carrier, so that they will have hidden alleles in the autosomal recessive carriers. So many people are walking around carriers for stuff that they have no idea because the disease hasn't manifested itself in them. So it's going to be twice the number of the incidence is going to be the number of carriers, and that's because they're heterozygotes, so they have one of the disease causing alleles, but you need both of them to actually show the disorder. Does that make more sense?
And you're right, just to add on that, it is allele frequency, right? You're still assessing for Q, which at this point is the disease causing allele, because we really don't care at that point if they're, you know, if they're autosomal recessive, right? We don't care if they have the big copy. We know that they're a carrier, but what we need to know is what are the chances of these carriers existing, basically the allele frequency that exists at this given time.
Yeah.
so we're looking for the allele frequency of the carriers.
Yes.
Okay, thank you.
Yes, if you're solving for the Q is going to be the allele frequency. Yes, like at the beginning, whenever, you know, there's that table, and just reminding to remember that there's 2 alleles on each person. So they could be 2 normal alleles, 1 disease causing allele and 1 normal allele, functional allele, or 2 disease causing alleles. So you have to think of them as separate alleles, but remember that people are going to have 2 of them. And that's kind of with the autosomal dominant down here, Kishore. That kind of makes a little bit more sense because the autosomal dominant is going to have 1 of each because they're heterozygous, but they are going to be affected. So with these, the 2PQ is going to equal the incidence, which you can again cancel out the Q. So the P. So the Q is going to equal half the incidence, and that's because the Q is the allele. So you're going to have that many alleles. Half of the people that are affected will have half. So the number of affected people, half of those are going to be the disease causing allele, because you only have to have 1 to be affected.
Yeah, and this kind of makes intuitive, sorry to jump in, it kind of
No,
makes some intuitive sense with the autosomal dominant, because remember what Dr. Sobring said is that you very unlikely you're ever going to see someone with 2 of the same disease causing alleles, because most of the times those don't have either fetal viability or they have high mortality rate. So you wouldn't be able to assess for this. So typically with most autosomal dominant issues or disorders, you're always going to see big A, little a, etc. Because if it was big A, then that's problematic.
yeah,
Thank
for
you.
sure. I think he even gives like that example. He had us calculate one of them where you would have to calculate like a hypothetical of someone being homozygous for the autosomal dominant disorder. And that's when you would square the Q and you'd get like some really, really crazy number that's like, it's very, very low probability. So that just builds on what you guys learned in FTM 1, where someone homozygous for an autosomal dominant disorder is going to be extremely rare, like negligible as far as calculations. So same thing here. So the incidence, again, the incidence of the disease is going to be the people that are affected. Here, you're going to be thinking about the heterozygotes because of what Kishore was just saying. You're not going to have those homozygotes. I mean, this would technically include heterozygotes and people that were homozygotes, but you're not going to have a number of homozygotes that's going to matter because it's going to be so small and so rare. So this is going to be, again, this is just reiterating that Q, the disease causing allele, is going to be half of the incidence. The number of disease causing alleles is going to be half the incidence because you only have to have 1 Q and then you can have 1 functional allele to be affected. Yes, and then X-linked. So remember that men are hemizygous with X-linked disorders. They only need to get one copy. They're only going to get one copy from their mother. So if it's the affected one, they're automatically going to be affected. So in this case, Q is just going to equal I, which is, in my opinion, these are the easiest ones because it's just Q equals I. The incidence, the number of affected males is going to be the disease causing allele because they only get one of them. So that's why it's going to be the same as the incidence. Now, they'll probably more often ask about the female carriers in the X-linked ones because it is pretty simple that Q equals I, so the incidence is going to equal the Q. I think the next slide has this female carrier. Yeah, so in order to get that, you're just going to double half the incidence. Yes, half the incidence will give you the Q. But when it's in fractions, you have to remember that everything's in fractions. So like you're going to be multiplying basically the denominator, which is why it can get a little bit tricky. So yeah, but they're almost always going to probably ask you about the female carriers, the heterozygote carriers with the X-linked recessive, and you're just going to need to half the incidence, and that's because of that allele being, they're going to be carriers of the allele not affected.
I have a question.
Okay.
How does that apply? This whole thing, I think I wanted to ask Dr. Sabarin about it, but how does that apply to X-linked dominant? Like, what about if we have a question about X-linked dominant?
You won't. I'm pretty sure you don't do X-linked dominant in Hardy-Weinberg because I don't think.
Yeah, because then this one is only male from female. So like the males are not affected, so it's not male from male. So we wouldn't have a question like that.
Yeah, I think for population genetics, you're only going to need to focus on these 3 inheritance patterns that he has given you in the lecture, as far as the specific inheritance patterns and how to apply the equations for those. And I know he has that slide where he said something about if each of these had an incidence of 1 in 25,000, which would have a higher number of carriers or of affected people or heterozygote carriers and things like that, just to demonstrate how the numbers work. And if it doesn't get covered in lecture, then you don't need to worry about it. I know there will be different types of questions on like the exceptions and things that could be exceptions to the rule, but I wouldn't worry about X-linked dominant if you guys didn't cover it because we didn't go over that either.
You do need to know the disorders associated with it, but you don't need to know it based off of Hardy-Weinberg and calculating. So
yeah, exactly.
Just be aware of that.
So anything that you learned in FTM1 with the X-linked disorders, yes, that's still fair game. But as far as applying it with this, no, you don't need to worry about that.
Okay, great. Thank you.
Yes, of course. So these are, I just memorized these just to, I mean, it does make sense, like Kishore was saying, it makes intuitive sense, but it gets a little bit tricky with the fractions and stuff because you're either dividing or multiplying and it's fractions. So you have to remember like, or in the squares, squaring and square rooting, the fractions can be a little bit tricky. So just make sure that if you are having a hard time with it, I'd recommend just memorizing these because it helps and you don't want to spend extra time. This exam specifically, I would say was probably the one I was most likely to run out of time on just because there's a couple of areas where there's math between this and the pharmacology stuff. So you don't want to be wasting time trying to remember equations or remember which one you're supposed to be applying. You want to just know it, do lots of practice problems, know these equations, and then that way it'll be kind of second nature. This was just put in here for just to show you, as he already wrote here on the slide, you don't need to memorize this at all. You just need to kind of recognize that there are certain trends amongst different populations. The perfect example that he likes to use is the Tay-Sachs disease amongst the Ashkenazi Jewish population. And that's because there's not many in the population in the first place, but they all like mate with one another. So you're going to have a much higher rate of carrier frequency or basically any possibility for that disorder. But in comparison to the rest of the world, theirs is like way higher. And that's the only population that you see that in. So it just depends. But like if you'll look at this, you just need to kind of know the general trends and understand how that works in comparison to the rest of the population. But this is not something you need to memorize at all.
Sorry, I have a quick question.
Okay.
Would you be able to go back to the previous slide where you have the formulas for each one of them?
Sure.
So like I'm just looking at the slides that he gave us, and like in one of the examples, it was like the frequency is 1/10,000, right? And then once you calculate the Q would be 1/100, right?
For
And
autosomal recessive?
correct,
Yeah,
autosomal
okay.
recessive, right?
Yes. Uh-huh.
And then after that, for the carriers, he just multiplied that number by 2, which 1/100 multiplied by 2 would be 1/50, right? So then, but here you guys, the formula that you have here, it might, like if you multiply 1/2 by 1/100, oh, it would still give you the same. Oh, that's interesting. Okay, awesome. Thank you. Sorry, I just was a little
I
bit
know
confused. But
it is
easier to memorize
tricky.
then.
I completely understand because I had the same problem. A lot of our class had the same problem, but if you actually work it out, you will get the same thing, but it just can be tricky because of the fractions and because of the square roots and because you're sometimes doubling or you're sometimes multiplying by half. So it can be a little bit tricky for sure.
Thank you so much. I'm sorry to take your time.
That's okay. No, of course, of course.
So for autosomal dominant and X-linked recessive for females would be the same formula?
Autosomal dominant and X-linked recessive for females. Yes, because if you think about it, the autosomal dominant is going to manifest itself in the heterozygous state. And then when you're looking at X-linked recessive carriers, you're also looking at heterozygotes. So that's why that would be the same. The X-linked recessive affected would just be equal to the incidence because those are all going to be affected, but the carriers would be a heterozygote, and that's why it would be the same as the heterozygous state you're thinking about with people that are affected in an autosomal dominant disorder. Does that make more sense? Okay, we'll move on. Okay, so where it all comes together is you still are going to have to know the 2/3 rule, the 1/2 rule, the risk in general of passing certain diseases based off of inheritance patterns to children. Like you need to know those Punnett squares and that, you know, there's a 1/4 chance of autosomal recessive and all that, but you have to take it a little bit further with the population genetic stuff. So keep this in mind, keep the mom times the dad times the Mendelian frequency, and then definitely keep in mind that 2/3 rule and that 1/2 rule. I know that's already been learned, but that will continue to be a thing. And as long as you have that down, you'll have a lot easier time figuring these out on your exams. I think we have some practice questions on these.
May I ask a quick question in regards to these rules?
Yeah.
I just had a question because I was solving a problem and I thought it was like the 1/2 frequency of like getting passed down to like what you have here, the 1/2 rule, but it was actually 1/3 because it was like 2/3 times 1/2. So I guess my question is like, when does the 1/3 come into play?
It depends on the type of question and what they gave you. I know I was going over some of these before y'all's FTM 1 exam, and there was one that was like that, and the answer was 1/3. So I know what you're talking about, but the way that they can word these can be super specific. So if you can maybe either find the question or we can talk about it at the end, then we can try to help go over that.
Yeah, for sure. I guess it is difficult without like the the inheritance pattern for sure. Yeah.
Yeah. And they do get a little bit tricky for sure. And that's also the case with some of these practice problems. So we did put some of the trickier practice problems on here, but also, and we'll get to this, but after these practice problems, Dr. Sobering for us specifically did these short, like 5 or 10 minute videos going over, he had a whiteboard and he went over all of the possible situations that he could give you on your exam, because there's this old video floating around that you guys might have already been recommended by other people to watch, and it's good, but he goes over problems that are more complex than he's going to give you on this exam, because when that video was made was when they were still taking the exams with whiteboards on the island. So they have adjusted how far out you're going to have to go, or how many generations you're going to need to know carrier status and things like that. So he actually last term made separate, short, quick, easy, separate videos for us They're super helpful. I went and rewatched them this morning, and like, not only are they helpful, they're hilarious because it's just Dr. Sobering and he's funny, but I've linked all of them here on one of these slides. So if you guys continue to have problems after we get past these, go watch all those for sure, because it makes it so much more clear, and he draws out the pedigrees and everything for you on his whiteboard. Okay. Just a couple of exception things, because I remember we did get tested on, this is like more conceptual stuff, but I think this probably was introduced to y'all when you first got introduced to sickle cell a few weeks ago, but when people are affected with sickle cell, actually, I think when you're a carrier, that's when you have the heterozygote. Yeah, of course, heterozygote advantage. So sickle cell anemia, so since it's autosomal recessive, you have to have both of the disease-causing alleles to be affected. But if you're a heterozygous carrier, there's shown to be some protection against malaria because of the way that it works with the red blood cells and stuff. And in CPR, you guys will learn a lot more about that. But just know that the heterozygote advantage thing applies to people who are heterozygous for a disease, but it can actually be protective over a completely unrelated disease. So that's this malaria and the sickle cell is a classic example of that you could get tested on. Founder effect. I remember getting tested on this one at least once. They'll give you like a situation of a population and show you some kind of trend or give you some kind of details about it. And you'll need to know the difference between founder effect and new mutation and genetic drift and things like that. So there's a DLA that you guys had that's exceptions to the Hardy-Weinberg equilibrium. It's very short, it's very simple, but it's got a few of these on there, and just make sure you know those, because again, it's an exception and exceptions are testable for sure. So know these. I know for a fact we had a couple of founder effect questions that like came back more than once. So just look those over. It's pretty easy. Just make sure you know the difference between those. Okay, we have a few practice problems on these to make sure y'all have it down. I think Kishore has a turning point set up and the session ID is right here. Okay, so it looks like most people chose D on this one. Is there anyone that would like to try to take a stab at this and see if they want to explain it or kind of walk us through? There's a few slides here that are going to walk us through it, but is there anyone that wants to try to explain this?
I can.
I can explain it.
I can.
Oh, I like it. I love the eagerness. Yeah, y'all choose who's going to go, but somebody try to explain this one.
I don't mind trying if
Okay.
anyone else doesn't mind. Okay, so I basically I looked through the prompts and like it says that the husband's brother has a daughter with cystic fibrosis. So based on the rules, we know that the husband's brother has to be a carrier, which makes him, the husband, have a half risk of being a carrier as well. And then the wife having a sister with cystic fibrosis means that that's the whole premise of the 2/3 rule, which we learned for FTM 1. So that makes her a 2/3 risk of being a carrier. And then the Mendelian frequency for the child being at risk is 1/4. So when you multiply that together, it's like 2 over 24, and then it simplifies to 1/12.
Perfect. Yep, that was perfect. Great, good job. That's exactly how you guys will have to be doing these too. So there's usually going to be 3 steps. There's going to be like 3 multiplications that you might have to do together at the end. And that's on top of already sometimes having to square root or square to find the incidence or the Q or something like that. But more times than not, it's going to be the mom times the dad times the Mendelian after you have already done your math. So yeah, get comfortable with these for sure. Okay, let's try this one. Okay, you guys are killing it. Hold on, Kishore, don't go down yet. Does anybody want to explain this one? This one's a little bit trickier. And we were just discussing this before we started.
I can try.
Okay, go for it.
They say the woman, she has a niece who has a cystic fibrosis. We know cystic fibrosis is autosomal recessive. Then she has a niece, that means half risk she's gonna have the disease. I multiply half, then the man, he is from population, let's say 3,600. Since it's cystic fibrosis, I will take the square root of the incident to get the mutant allele. Then I will divide that by 2 to get the affected, the carrier. Is that right?
Yes, yes, that actually is great. That's a better way of explaining it than
When I took the square root, I get 1/60, then I divide it by 2, I get 1/30. And the Mendelian frequency for the child, it's 1/4. And I multiply them all and I get a D.
Okay, good, great. That's really good.
And here on the slide for the mother, it says 1/4.
Yeah, so it is, and that's because we actually were just going over this before this started because I was like, this question's tripping me up. So it says that the niece is a carrier for the mutation. Now, I'm pretty sure that you guys are not going to get asked questions that are this far out. It's definitely fair game to say that the niece is affected with cystic fibrosis, and then you would know that the mother would be 1/2 in that situation. But the problem is here that the niece is a carrier. So you have to pick up on that. And then Sarah is the one that explained this to me because I seriously was like, this is tripping me up. I couldn't like conceptualize it. But if you think about it, so whoever the sibling is of the woman, the proband here, the 27-year-old woman that's coming to learn about her recurrence risk, you know that one of her siblings is going to be either the mother of the father of the niece that they're talking about, that's the carrier. So you would think 1/2 because that's like the 1/2 rule, except you have to multiply 1/2 by 1/2 because you're not sure if the niece got the the allele from, you're not sure which parent the niece got the allele from. So you have to multiply it by another 1/2. And so that's why the mother here is going to be 1/4. But I also had a difficult time with this. So I understand why you guys are thinking this. And I also don't think you're going to get a question like this. And that's probably why you're thinking 1/2, because whatever you're taught is what's going to be on your exam. And I think this one's like a little bit too far out. And then also in those videos I was telling you guys about with Dr. Sobering doing the examples, I watched them this morning and he didn't give one that was like this. So I wouldn't worry about that. But yes, you're right to think about the 1/2 rule. I think the difference here is just that the niece is the carrier for cystic fibrosis. She's not affected. So your logic was, most people got this right. And so your logic was right. So just, you guys are on the right track there. Don't stress too much about this one.
Would you
Sorry,
please?
I have a question on the previous one.
Okay.
I understand the dad that is 2/3 and the kid's risk is 1/4. I just don't understand why the mom is 1/2.
Can we go back up a couple of slides, Kishore? Are you talking about the first question?
Yeah.
Okay. So this is going to be kind of like more the logic that we were just talking about for the second one, but the husband's brother has a daughter that has cystic fibrosis. So the husband is going to have a 2/3 chance of being a carrier. And then the wife has a sister who does have cystic fibrosis. So wait, I think this is backwards in order.
Yeah, the order is off.
The mother should be 2/3 and the father should be 1/2 here, right? Is that what you're gonna say, Kishore?
Yep, that's what I was going to say. If I see a couple of folks who had issues with this, let me go back up a couple of slides so that you guys can take a peek. Make sure you're well aware of these two statements so that you guys can be very confident on when a sibling with the disease and then you having the chance of becoming it or being a carrier status is 2/3 here, right? Whereas if its sibling is a known carrier, then you have a 1/2 risk, okay? Just be really solid on those two.
I see what you're saying though about that one, just because of the way that the explanation was put on that slide. It looks like it's backwards because yes, the mother is going to be the one that's 2/3 here because she has a sister that has cystic fibrosis. So that's that 2/3 rule we learned back a few weeks ago. Now the father is going to be 1/2 because the father's brother has a daughter with cystic fibrosis, which means that the brother has to be a carrier because it's a recessive disorder. So both parents have to be carriers in order for the child to be affected. So there's a 100% chance that the father's brother is a carrier, and that gives him a 1/2 chance of being a carrier. So the father is 1/2, the mother is 2/3, and then the Mendelian is the 1/4.
Okay, thank you.
Does that make more sense?
Yes, definitely.
Okay, cool.
Before we kind of go further, I will highly suggest you guys do a couple of practice questions with these and memorize the Mendelian frequencies or the incident rates for autosomal recessive, dominant, and etc., because it will help you get the quick math out of the way, because they never mentioned in this vignette that it was a autosomal recessive thing, right? It's cystic fibrosis. You should know that from FTM1 that it's a kind of a building of your knowledge base. And based off of that, remember it's autosomal recessive. So the chances of the child actually getting it based off of a parent who has big A, little a, and the other parent having big A, little a is 1/4, okay? Draw upon it if you need to reinforce the idea, but I highly recommend memorize those because it's gonna give you that extra couple of seconds during the actual exam.
For sure. And this is the exam that you'll need those seconds on. Also, like the more practice questions you do, the easier it'll get, the more it'll make sense. That's the case with anything really, but these specifically. And then, yeah, for sure. We have a couple of more just to go over some different types of.
I have a quick question.
On the question, when it say she has a niece who is a carrier, do we get 1/4 because she is a carrier? She get 1/2 risk from the dad, 1/2 risk from the mom. Then we multiply them, we get 1/4.
Yeah,
So
basically, yeah. But yeah, go ahead, Sarah.
actually, you don't know. So this is going to be the 1/2 rule. It's actually the next question, Kishore, right?
Yeah.
Yeah, so for this one, you have a 1/2 chance of the gene coming from the mom or the dad of the child that's a carrier. So if you're trying to figure out the sibling of that parent, you have to do the 1/2 rule, because if your sibling is a carrier, then you have a 1/2 chance of being a carrier, right?
Yes.
And then you would also have to multiply that again by 1/2, because you don't know if this recessive allele came from the mom's brother or sister or the other parent. So you have to take that into consideration. Just because it says carrier, that's why we had to multiply it by an extra 1/2, because we don't know if it did come from that parent or the other parent.
Thank you so much. I highly appreciate it.
No problem.
Yeah,
You guys
don't worry too much about that one. Yeah,
Yeah,
don't worry
if
too
you
much about that one. I don't think you're gonna see one like that, and it is super complicated because yeah, like I said before this, I was like, I don't understand this question, so don't worry about that one too much.
Okay, one more thing before we kind of move on. There was a couple of other questions, but Morgan, Liz, can you please share that the text that you just sent me directly with everyone in the chat? I think that's a great way, and you've summarized it perfectly, and I think everyone would benefit looking at that. Also, we had a question on how do you figure out the father from the incident, right? Like the frequency. Cassidy, could you take that question?
Yes, just asking about like the incidence rate and the father's yeah, of course. So this is going to be autosomal recessive. So this is when the Hardy-Weinberg stuff and the population stuff comes in. So the incidence in the population is 1 in 3,600, which means you're going to need to take the square root of that. And then the square root of 3,600 is going to be 60. So that's going to give you that 1 in 60 would be your father. I mean, no, not the father. That would be the Q. So then you're going to have to multiply that by 1/2 based off of our rules with being a carrier and the 2PQ and the 2Q and stuff like that. So it's going to be 1 in 30 is going to be the father's risk, because you're using the population. You're no longer using, he doesn't have a family tie to this. So anytime somebody doesn't have a family tie, but they're in the population, they're going to give you this population incidence. And that's when you'll have to use that as far as the chance that the father is going to pass on. So that's why it's going to be 1/30 for the father. And then when you multiply all of those out, you're going to get that 1 in 480. Does that make more sense? Okay, cool. Okay. This one kind of makes you think a little bit, but let's see how you guys do on this one.
Also, for anyone who came in a little bit later, we are using Turning Point. The session ID is in the bottom right corner. Feel free to click it.
Okay, so it looks like most people chose answer A, but there is a little bit of a split. Is there anybody that would like to explain this one?
I can go for it. Go ahead.
Sorry.
do this one.
Okay,
So for each parent, so with the dad, his mom has the mutation for the 4, and the mom, like the stepborn parent, the mom, her mom has the mutation for the 3. So since it's only like one parent is heterozygous for each mutation, each parent then has 1/2 chance of getting that that allele, and then their child has a 1/4 since they're both heterozygous of having that. So it's a half times a 4th.
Yep, great job. This is just another way of showing it to you like on a pedigree. It's a little bit more complicated because you have to think a little bit with the whole way that they laid this out in the genes and the mutant genes and things like that. You just, like she said, you just got to look and see what they're pointing to there. So you're looking for the DR4 and the DR3. And yes, exactly. That was explained perfectly. I think there is one more question on these. Yep, okay, let's try this one. This is the last genetics question. Okay, you guys are doing awesome. This is great. Is anybody that would like to explain this one? This one's a little more straightforward.
Well, this one is saying that the allele frequency is 1 out of 400. So it's asking for the frequency of homozygotes. So that's Q squared. So that's 1 over 400 squared, and that's D.
Perfect. Excellent. Yes, and then, yeah, this is just a follow-up. So if we were asking for the number of carriers, which the answer is already here, you're just going to do the 2Q or 2PQ, but without the P. So you're just going to do 2 times that 400 that was given to you in the problem, and that would be 1 in 200 people are going to be carriers if there's an allele frequency of 1 in 400. But yes, that was great. Good job. Y'all did good on those. Okay, here's the links to the videos. You should just be able to click them from the slides. Hopefully, let us know if they don't work. But like I said, they're all pretty short. All of them I want to say are like 10 minutes or less. They're excellent. He explains them super well. You'll also have some laughs out of them because he's hilarious and like accidentally inhales some of his whiteboard spray. And also he does them all at the same time. You can tell because he's wearing the same shirt, but he changes his tie for every video because he's just, you know, silly like that. So I would definitely recommend going and watching all these after this if you have time, because they'll make the concepts a lot easier for you. And he does like for each category, he'll do any of the possibilities of the way that the question can be asked. So these are excellent. Definitely watch all of these. I will probably watch them again just because they're funny.
You think that you could go over again like the D3, D4 question one more time? Sorry.
Yeah, for sure. Yeah, that one is a little bit trickier, but that's an example of a way they could ask you it. Because in my opinion, that one just looks, it's one more up. And that one just looks more confusing than it actually is. So what you have to think about here is, so if you're looking at the first generation, it's showing you the alleles that these the grandparents of the proband, the person in question, the alleles that they have. But the first sentence of the question is telling us that the ones that we're looking at are going to be the DR4 and DR3. So only one parent of the proband's parents, so the grandparents, is going to be heterozygous even for this disease causing allele. So there's going to be a 1/2 chance that that is passed down to either of the parents, because there's one on each side. And then just the 1/2 times 1/2 for both of this person's parents is just going to give you 1/4. I mean, I'm sorry. I'm so sorry. I misspoke. There's going to be a 1/4 chance because it's autosomal recessive. And then so all of those multiplied together would give you the 1/16. I apologize. I misspoke there.
Okay, thank you.
Yes, you're welcome.
And we had one more question for this last question.
Okay.
Why did we why didn't we just square root the 1 over 400?
I'm not sure I understand the question. Are we talking about for the carrier status or for the actual question?
Oh, go for it. The person
I
who
think
had the
maybe
question.
like 1 in 400. We just have to know that that's the allele frequency, so that we have to actually like not take the square root but square it. Or sorry.
Yeah. Yes, you're on the right track there. So that's the thing about these, is they can either give you the incidence or they can give you the Q. So here you're already given the Q, but you need to find the I. So if you think about it, there's going to be a lot less people that are affected than there are going to be that have the allele, because to be affected, you have to have both of them. So if there's a frequency of the allele of 1 in 400, then the people that are actually affected by that and have both copies of it is going to be when you square that. So you have to be able to interchange those. So you have to know how to go from I to Q and Q to I, which is going to be the opposite of one another. That's why it's best to do a lot of practice with these, because it really can get tricky. Does that make sense? I don't know who asked the question, but yeah. And then also, like, if you just think about it logically with these, there's going to be less people affected than there are carriers. So if you ever get stuck and you think, okay, well, there's not going to be 1 in 5 people affected with this if there's only 1 in 2, like, you look at the carriers versus the affected people, and you should be able to at least rule out some of your answers there. Okay. If there's no more genetics questions, we are going to move on.
Okay, gene regulation. Let's go ahead to the next slide, Kishore, please. Okay, lac operon. So I know that lac operon, most of you guys have probably learned it in undergrad at least once. For this, it's just important to know which enzyme each of these genes is going to represent. So you have like your LAC-Y, which is your permease, I believe, and then your beta galactosidase is your LAC-Z, and then LAC-A is transacetylase, I want to say, but someone correct me if that's wrong. So you guys just got to kind of know what they stand for. And then also your LAC-I is separated from the lac operon genes. And the function of your LAC-I is to pretty much make your repressor protein. So LAC-I encodes the repressor protein. It's going to make that repressor, and that repressor is going to come and bind to your LAC-O. And wait, Kishore, can we actually stay on that slide? Because I'm going to explain it all. It's going to bind to your LAC-O and it's going to block transcription. So there's a few moving parts to the lac operon that you kind of have to keep track of. And once you put it all together, then it starts making a lot of sense. So the two situations that you're trying to figure out are, is glucose present and is lactose present? So with your lactose, that one will come second. So for glucose, if glucose is present, then you have decreased cyclic AMP. If you have decreased cyclic AMP, nothing is binding to your cap protein. Your cap is going to not be inhibiting anything, or it's not going to be attached to anything. So pretty much if glucose is present, you don't get your lac operon transcribed. And that's something you have to always keep in mind, because even if you do have lactose present, you still can't transcribe your lac operon if glucose is also present. So let's move on to the next slide really quickly. Okay. So again, if you have no lactose, then you will have your lac-I making your repressor. Even if you don't have glucose, if your lac-I is making your repressor, but you have no allolactose, which is made from your lactose, to come bind to that repressor at your lac-O site, change its conformation and make it leave the lac-O site, then you're not going to be able to transcribe that lac operon. And let's move on to the next one. So again, if you have no lactose, this is the situation. You don't have anything to get rid of this stop sign over here. This stop sign is going to represent your repressor. So if you have no lactose, you're not making allolactose. Allolactose is responsible for binding to that repressor protein, changing its conformation so that it no longer can sit nicely on that lac-O spot. And so it'll force it to leave. But if you don't have lactose, you don't have the allolactose. So your repressor is just chilling right here at your lac-O. None of your transcription machinery can move. Does that make sense? Yes. Okay. Okay, so again, this is just kind of repeating that. Let's move on. I think there's a summary slide that just kind of makes it look better. So let's stop here real quick. If you have no glucose, then this is the kind of cascade that you want to keep in the back of your head. No glucose means increased cyclic AMP. Cyclic AMP is your hunger signal. So you have the hunger signal in the presence of lactose, but you also need no glucose. So your cyclic AMP binds to this active CRP or CAP, depends on what diagram you're looking at, they'll be named either one. And then that's going to bind to the CAP binding site. And then when you have your lactose, your allolactose is chilling over here, bound to your repressor so that your repressor can't bind to lac-O. So no glucose and lactose is present, then you are going to have the promoter bound and nothing repressing the operator. So you are transcribing your lac operon. And let's move to the next slide. And then I really used this table when I was trying to put it all together. I just kind of took this table and wrote out all of the detailed notes around it for each of these squares, so that if they give you any situation, whether you have or don't have glucose, or you have or don't have lactose, you can figure it out pretty quickly. So just keep like practicing with it. I honestly just explained it out loud to like myself and my friends, and it kind of sticks after like the first couple times. Okay, let's move on. Okay, and then again, this is just a first aid slide that kind of goes into this again. Low glucose means increased adenylate cyclase, which increases your cyclic AMP, and that activates your CAP, and it's going to increase transcription of your lac operon. And then when you have high lactose, it unbinds the repressor protein from the operator site, and that's your allolactose that's going to be doing that binding. And then that is also going to increase transcription. So you need both of these parts to be present in order for you to actually transcribe the a functional lac operon. So that one was kind of given to you as an example of gene regulation in prokaryotes. And then there was the rest of the lecture, and I believe a lot of the DLA that was giving you more examples of gene regulation in eukaryotes. So when we're talking about cis and trans regulatory proteins or regulatory factors, your cis is your DNA sequence. So when they ask you what your cis regulatory elements are, they are these purple boxes, this red box, these purple and blue boxes, like it's actually the DNA sequence. And keep that in the back of your mind, because the things that bind to your cis factors are your trans factors. Okay, let's move on. So for trans regulation, these three terms you should be very familiar with. I believe we were asked a couple of them on the exam. So competition is just when you have the repressor binding to the enhancer, and it's gonna pretty much like competitively compete with the activator for that same binding site. So you're going to get decreased transcription because at some places the repressor will bind, at some places the activator will bind. So depending on what ends up getting that spot. With quenching, the repressor is going to bind to the activator, and it's going to block the ability for it to do anything to bind to that site. So that is going to have no transcription possible because it's kind of just like sitting there and nothing else can bind there. So nothing is happening. With blocking, sorry, can we go back? Oh, yeah. I can't see the slide. Okay, thank you. With blocking, so the repressor is going to bind to the activator protein's activating domain. So with your blocking, that's where you have that activation domain sitting on your cis sequence, and then that repressor is going to bind to the top of that, and it's going to interact with the transcriptional machinery. So you're just looking at different ways that can regulate whether gene expression is happening or not happening when it comes to your trans regulation. Let's go to the next slide. Okay, with
I'm sorry,
your...
I have a quick question. Actually, I want to clarify. In blocking, when the repressor bind to enhancer sequence, it will completely block it, so we won't have any transcription, correct? Okay, perfect. Yeah. Thank you.
So that's going to be blocking. Yes, it's going to prevent it from even interacting with the transcription like factors and transcription machinery. So you're not going to be able to initiate or do any transcription.
Exactly. So even though if the binding happened, but the transcription would not take place because it's completely blocked. So they cannot interact with each other anyway.
But there will always be some basal level transcription going on because it's still at the basal factor is still attached and they're doing this. The enhancers just make it more.
That's going to be for your enhancers. For blocking, yes, if the transcription machinery, like you can see in that in the image, if the transcription machinery is able to assemble, then you're always going to have that basal level of transcription happening. And again, yeah, you can either up or down regulate that based on what is binding or not binding.
Here, I'm going to jump in quickly and add a little bit more detail. If you guys think about it, this is like a molecular machinery, right? Like it's kind of like a zip. It just keeps going back and forth and creating transcripts or like copies of your genetic makeup, your mRNA that eventually gets turned into the protein. So it's just a constant repeat sequence. That's what the enhancer essentially does. It kind of swivels on itself and binds to this basal machinery and allows it to just constantly make the same transcript. But when you have this kind of blocking mechanism, you will have basal level products being made, but it's not going to be enough or like sufficient amounts to have like any meaningful, impactful change. Okay.
Yep. Okay, let's look at the next one. So for your hypoxia inducible transcription factors, this is trying to show you an example of a coordinated gene regulation. So you have this one gene that's supposed to coordinate regulation of a bunch of different genes in different locations in the body and in the cell. So pretty much what's happening is you're looking at hypoxic conditions. So either oxygen is present at sufficient levels or it's not present at sufficient levels or not present at all. So when we're looking at this transcription factor, your HIF1 alpha is going to move into the nucleus and it's going to dimerize with your HIF1 beta. And once those two are dimerized, they're going to bind to your hypoxic, what is it called? Response element, or your HRE, and that's going to either cause transcription or not. So when you have a situation where you have high oxygen, then you don't need your hypoxia inducible factor because you have high oxygen. You only need this when oxygen is not at sufficient levels. So what's going to happen is these prolyl hydroxylases over here are going to, they're going to pretty much hydroxylate the prolines that are on the HIF1 alpha. Once those proline residues are hydroxylated, it's pretty much tagging them for degradation by the peroxisome. So when, or the proteasome. So when you have high oxygen, you're pretty much telling your body, I don't need the hypoxia inducible factors because I have no hypoxia going on, and it just degrades it. When there are low or no oxygen levels, then your HIF1 alpha is going to be stabilized. It's going to move into the nucleus, like I said, dimerize with that HIF1 beta, and then it's going to activate gene transcription and regulation for a bunch of different genes that are going to try and correct that low oxygen level. Let's move on to the next one. So when we're talking about your glucocorticoid receptors, those are also going to dimerize. When cortisol is present, then your zinc finger DNA binding domain is going to be where those cortisol are going to bind on the glucocorticoid receptors once they're dimerized. And then that whole complex is going to interact with activators and enhance transcription, or they can also interact with repressors and decrease or downregulate transcription. So on the next slide, you can see that. Again, so it depends on what is in contact with that dimerized glucocorticoid receptor. Let's move to the next slide. And then the other gene regulation concept that you guys were introduced to is your idea of microRNA and your siRNA. So with this whole complex right here, we're pretty much talking about how we can either permanently inhibit transcription or how we can temporarily inhibit that transcription. So you have these long PRI, not pre-miRNAs, and those are going to be processed into these hairpin structures over here. And then drosha is going to come in, and it's going to pretty much, it's going to create these, it's going to create these hairpin pre-miRNAs. Then exportin is going to come in and do exactly what you think exportin would do and export that hairpin structure. So dicer is going to come in next. It's going to dice it up, and you're going to get single-stranded miRNAs, and it's going to create this thing called a RISC complex. So your RISC is pretty much going to be your 1/2 of that hairpin structure that's already been cleaved. And then when we're talking about your miRNAs, it's going to bind imperfectly to whatever you're trying to inhibit. So the 3' untranslated region of that target mRNA that you're trying to inhibit, it's going to bind to it. And because it's imperfect binding, it's not permanently turning it off. It's just turning it off while it's bound. So it's blocking that ribosome from being able to translate, but it's not causing any permanent degradation to that sequence. And then for the next one, your siRNA, it's pretty much going to start with your double-stranded siRNA, just like you started out with your miRNA. But the only difference is it's going to do the same thing where it's going to become single-stranded. It's going to make that RISC complex, but this is going to perfectly bind to that mRNA sequence that you're trying to either inhibit or destroy. And because it's perfect complementation, that's when you can say that it's going to have endonuclease activity, and that's going to get cleaved and destroyed. So you're never going to be able to unbind it and translate that sequence because you are degrading it. And that's honestly the only real difference that you guys have to understand between the miRNA and the siRNA. Just know that one of them is permanently inhibiting and one is temporarily inhibiting. And then also just know what these little enzymes do. Exportin is exporting, dicer is separating two strands, drosha is making that pre-miRNA, and that's honestly as much as you need to know for this one.
Jump in. Oh,
I'm
sorry,
sorry,
Scott.
Michael?
Sorry, one of our questions kind of also highlighted that siRNA was like an exogenous double-stranded RNA, whereas microRNA was endogenous single-stranded. I don't know if that came up on the test or not, but that was kind of a confusing little caveat.
It didn't, but when you look at your miRNA, because of this hairpin structure and this little imperfect binding site over here, that's why it's like when you look at that little loop, it's not perfectly binding. So the main idea is that miRNA can be removed and you can resume translation of that mRNA, that target mRNA. So you just want to understand that like main concept that miRNA is temporary and can be removed. It can also end up being degraded or never be unbinding from that site, but you also do have that capability of the miRNA unbinding and just resuming whatever transcription or translation you were doing before.
We didn't have a question in that level of depth, but I just want to kind of flesh out your guys' knowledge and say like, our body just does not like double-stranded RNA. So whenever it sees double-stranded RNA, it's assumed to be foreign and it's typically destroyed. So it's more applicable. I think the vignette that we were given in one of the question stems from Sakai or during the quizzes, weekly quizzes, was that it was something to dealing with viruses. So just be aware of that if you see something like that. Most of the time, they are going to just ask you about like these, drosha, exportin, and dicer.
Okay.
Would you please one more time compare the siRNA with the miRNA?
You can actually, Kishore, go to that slide that you just scrolled up from, and I believe there should be something here. Yeah.
Perfect.
So with
Perfect.
with your miRNA, like Kishore said, this one is going to be mostly endogenous, and it regulates their expression by repressing mRNA translation, but not completely destroying that mRNA transcript that you have. With your siRNA, it's going to degrade that mRNA. And that's like one of the biggest differences you have to understand between these two. And then it's also going to be derived from double-stranded RNA, but so is your miRNA double-stranded pre-miRNA. So they're both starting out double-stranded, and both of them are going to be separated by that dicer before they go and bind to their target mRNA. And then you just should probably know that your siRNA is often exogenous. So like Kishore said, viral, or it could be injected.
Thank you so much.
No problem. I hope that made sense. I know it's like kind of dense and it was like a DLA, but I thought I'd try to explain it because you don't want to miss out on easy points from these questions because it's really just knowing functions. All right. Let's try a question. Okay, so most of you guys got this one. And when we're talking about Mick and Max, I'll explain that one in the next slide, but pretty much for this question, you just want to know that Mick and Max have to form a heterodimer in order to have the cell cycle progress. If Max is making a homodimer with Max, then it's not going to increase your gene expression or your cell cycle progression. Let's move to the next slide so I can try to explain that. So here you can see that when Mick isn't present, Max is just going to form a homodimer with itself, and it's going to repress gene transcription because it doesn't really bind that well to this enhancer DNA sequence. So if Max is desperate, Max will form a homodimer with itself, but it prefers when Mick is present, because when Max and Mick form a heterodimer, then things start moving smoothly and you end up getting your transcription, or your cell cycle will progress. And let's go to the next slide. So here we can see that when Mick is there, then Max wants to bind to Mick and form a heterodimer. And yeah, that's exactly how I looked at it. Actually, I just kind of created this whole relationship in my head, and it totally carried me through the exam, and I still remember it. So if that works for you, I definitely encourage that. So here you can see that your Max and your Mick, when they get together, they form this like perfect pair. They bind to that enhancer and they cause cell proliferation. So it's just multiplying. And then Mick can't do anything on its own, but Max can technically do stuff on its own. It just would prefer if Mick was around, so it didn't have to do it all by itself. That's pretty much what you have to know about that. So we know we forgot to give you guys a break last time, and we apologize for that. But we're gonna give you guys a short break, and then we'll come back and finish the rest of this. So
Alrighty. Okay. Welcome back, everyone. I hope you guys enjoyed these flowers, courtesy of Dr. Trotz's wonderful photographic skills. And let us make our way to connective tissue. One of the topics that she taught us, and I had a blast learning it because it kind of tied in everything that we learned in FTM 1 with Marfans and other like collagen and connective tissues. So clearly the connective tissues have some issues. We're going to find out more about that, and you're going to find out more about it even more in MSK, but this is kind of a quick introduction and some of the more in-depth focus topics that you're going to get in your next exam. Okay. First thing first, embryonic connective tissue, right? We have essential connective tissues that form, and they basically serve a multitude of different functions. You guys know this, connective tissues are essential. They essentially are going to contribute to everything that you know in terms of nutrients being transported, holding together your organs, microstructures that when we're talking about whether it's in the renal capsules, like what they're made out of. So there's a multitude of functions. I don't want to bore you guys with too many things, but you guys can read this on your own time. And what I will say is you do need to know the mesenchymal cells, the ground substance that they make, hyaluronin. It's the gelatinous consistency to the matrix. And we definitely had 1 or 2 questions on the umbilical cord. And remember, these are very buzzwordy. So remember Wharton's jelly, and you can be quickly okay, there's high amounts of water. If there's kinking, it can cause poor blood supply to the baby. So some of the key things that you can take away from it is the buzzwordy concepts, but loose areolar connective tissues has a couple of other important features, right? There are a large number of cells as compared to fibers. And you can see here, they have a multitude of different things here, okay? You're gonna see adipose, you're gonna see eosinophils and lymphocytes, and all of the eosinophils and lymphocytes are all part of the immune system. But the collagen structures, and the fibroblasts and all of those things in the amount that are present is going to inform you where you are regionally and as well as cellularly. Blood supply in the loose connective tissue is rich. You'll get more on this later in MSK2, where you start to look at dense connective tissue, dense irregular connective tissue. So that's just a quick peek as to what's going to come up. But for loose connective tissue, just be aware it's nice, rich oxygenated blood supply. You can do a nice set of gas exchanges and nutrients, diffusion of hormones and other factors. Location, typically beneath most epithelial linings. You're going to see it because loose connective tissue can take a lot of impact, right? Like you can constantly move around without taking too much wear and tear. Now, the laminar propria of most mucous membranes, such as those in the respiratory, urinary, and digestive system, is composed of loose areolar connective tissue. Okay, awesome. Now, dense irregular connective tissue. Okay, fewer cells, more fiber. It makes sense because the orientation of these collagen fibers allows for them to take resistance in all directions. So you're going to find them a lot more in the organ, capsules, periosteum, and the dermis. Make sure you guys know the specific locations of these because they are going to help you differentiate what type of connective tissue you're either going to see on a histological slide during the exam, or just make the general assumption of like, okay, they're talking about the dermis, it's dense, irregular, not as much blood supply here, but it has a lot more fiber, AKA your skin can kind of is more malleable, right? Like you can press on it and it doesn't really like just slough off and just fall apart, okay? Now, something a little bit more stronger, right, in terms of connective tissue is the dense regular connective tissues. Specifically, you're going to see the orientations are going to go in one direction as compared to the irregular. You see it's kind of all over the place. And this helps you kind of distinguish very clearly where you're going to find them, right? You have a lot of tensile strength, tendons, aponeurosis, and ligaments. Remember, those are great, those are strong, but the problem is that they have very few blood supply to that region, so that healing of ligaments and tendons is very little. So everything that happens at that level is more fiber, and you can't just pass material to for quick healing processes. So that's why there's a lot of tendon and ligament replacements whenever athletes kind of get damaged. Components, fibroblasts, and tendons have tendinocytes, okay? And remember, type 1 collagen. We're gonna go over the types. There's a nice quick mnemonic to kind of remember them. It's one of the ones that we came across earlier. Before we go on too far into our little adventure with connective tissue, let's do a quick question. Giving you guys a little bit shorter amount of time so that you guys can click in. In the meantime, feel free to message us any questions. It'll be a little harder for me to answer, but feel free to message. Yeah, fantastic. Almost time. Feel free to click in. And let's see how you guys did. Awesome. We had a little bit of distribution. That's okay, because this is one of the unique staining techniques that you often don't remember directly off the bat. So for connective tissue, specifically elastic connective tissue, remember it's silver staining, okay? A lot of the other ones that you've seen are going to be touched on later. I think in our drive, we even have a nice summary sheet that was pulled from Sakai that shows all of the different stainings and when and where you would use them. And this is tying in right here. Elastic connective tissue. Yes.
Sorry, I just wanted to like say, it's asking which one is not used for elastic. So the other three
sorry.
could be used and then the silver wouldn't be. And I know this because I got this question wrong on our exam with the aorta. So definitely know those.
Sorry, that was my mistake. Remember special staining, Orson, Roserson, and Verhofst. Okay, those are the unique ones that we haven't seen before. Silver staining, I'm pretty sure you guys haven't seen them before. I barely saw them even mentioned during small groups. So locations, ligamentum flavum, vocal cords, and suspensory ligaments of the penis. Okay, just be aware of those. We got tested on these specifically.
I have a question.
Go for it.
Silver stain, we use it for reticular. Is that right?
Retricular, yeah.
Thank you.
And also fibers tissue.
I don't.
Fibers.
For the stains, they're going to be on that like summary sheet. They're going to be stated as like a bunch of different things that they can possibly stain. But I think what you guys should focus on is like that main thing that like silver stain, reticular fibers, like associate those two with each other. That way, like you don't have to like think about what else is possibly in this histoslide. You just think like, oh, this is silver stain. So I'm looking for reticular fibers. Like try to associate it in that way and it'll make it a lot easier because they can all possibly stain for like multiple things.
Make sure you guys look at that stain sheet. Cassidy's mentioning it in the chat as well as what Sarah said. It's important. I'm pretty sure I missed that question too because we had so many different staining ones and we barely focused in on this one even during small groups. So just be aware. Okay. I will send you guys a link after we end the review session. Okay. Let's do this one. Sorry about that confusion, guys, on the last question. My camera feed was covering up the word not, so I was like, okay.
Yeah, I remember that one because there was like talking about Marfan's or something. And I was like, oh, I got this. I know this. I know this. And then it showed the aorta, which we hadn't learned yet. And then it was like, what was the stain used? And I was like, are you freaking kidding me? And it was like Verhof's or something was one of the answers. And so just, you have to pay attention to the little details on this because they're not going to ask you about H&E or something easy like that.
Don't worry, I'll tell you guys the day when you guys no longer need histo, which is when you guys get to turn 4. So we get to be rid of all of your wonderful histology skills that you'll never touch again. Okay, which of the following characteristics applies to this image below? You guys went for C, and that is correct. Okay. Now, uniocular adipocytes is one large fat droplet. Okay, that's white fat. That's the fat that we store as adults. What is the other type of fat that we see in babies, guys?
Brown.
Brown fat.
Brown fat. And believe it or not, we're in term 2 and we learned the actual mechanism of why brown fat is so important. So a lot of the stuff that you pick up now is going to come back. It might feel a little irrelevant at this point, but it is one of the more important connective tissues for babies. Okay. Typical for white adipocytes is a large signet ring. This is very buzzwordy. So this is a signet ring. It's one of those old school rings that they usually give for like when you graduate high school or something. And they have a large fat inclusions and organelles and other things are pushed to the periphery. So if you see this like washed out space, remember when they're preparing these slides, fat and proteoglycans and glycoproteins all get washed out. So you can imagine these large empty white spaces are perfect examples. And then their main energy source is triglycerides, right? We will cover more about those when you guys make it to term 2. and they have a nice rich blood supply. They will need to because hormones and other things are going to act on adipocytes. Okay, let's take a quick peek at adipocytes, brown versus white. You can see the nice clear examples, and you can tell quickly, right? Like you can see like the multiple like little droplets. Those are small, and they have, you know, its primary function is energy release, whereas this one is fat or energy storage. Okay, just be aware of that. Keep those two very separate, and you'll be a-okay with your adipocyte. Let's do this one. Yeah, our drive has a lot of things, and even links out to other drives and locations. Just be aware, if you guys have a hard time finding any of those, feel free to message us and we can kind of guide you guys to where things are. And trust me, if there's something that you come across that you feel like that could be on a drive, just message us the name of the document and we could probably find it in the drive. Okay. And when we cover farm a little bit in advance, we'll upload a nice summary sheet or document that's from another DES page that has everything on there that you need to know for farm at this level. Okay.
I'm sorry, can you repeat what you just said?
Yeah. When we get to the tail end of our review session, there's a lot of farm things that we're going to go over: pharmacology, pharmacokinetics, and pharmacodynamics, and everything. Those are kind of nicely summarized in another PowerPoint that was put together by a DES group, and we will upload one of those to our drive, and that'll help you guys a lot. It helped us when we were going through the material. Awesome. So you guys picked D, and that is correct. So a researcher is studying connective tissues and notices presence of cross-links which stabilize the collagen and elastin. Which amino acid is modified to allow for such cross-linking? It is lysine. What's happening with lysine here, guys? What does lysine do?
But you mean like it gets hydroxylated and glycosylated and all that stuff, and it helps with cross-linking, right?
Correct, exactly. And we're going to go more about that in a hot minute, but that's exactly what I was looking for. Great job. Okay, before we get into that, let's kind of quickly go over your guys' connective, or sorry, not connective tissue, but collagen. This is pulled directly from first aid. The way, literally when we started the term, I was telling Sarah, the way I remember it is type 1 collagen is bone, has the word 1 in it. So that's very important for osteogenesis imperfecta. Type 2, it's cartilage, right? So that's how I remember it. It includes hyaline, and you're going to learn more about hyaline cartilage in MSK as well. And then vitreous body and the nucleus pulposus. These are important. These words might seem like there's some far out magical spell, but they're not. They're going to come back, and you're going to learn more about them later. Then there is 3, retricular 3, and the pathology that they associated with is Ehlers-Danlos. We're going to cover that with you guys. It's a connective tissue disorder, and the key hallmark sign that you're going to see is with stretchy skin. In a hot minute. Then there's type 4, which deals with floor. So basement membrane is, I guess, the floor in this case. So if that helps you, I always remembered once I get to retricular, everything after that is basement membrane. So you've gone from levels and you've gone to the lowest most level. Awesome. Let's get this party started with this. Okay. Now, collagen has a very unique structure, right? But it's going to be dependent on the uniqueness based off of the region or the tissue that you're talking about. But it all has the same origin, right? The same thing that you guys learned from FTM1 and the start of this term, right, is in terms of post transcriptional modification is going to be critical, as well as post-translational. Okay, just be aware of that. Let's kind of go through it step by step. Where is this happening? It's happening in your fibroblasts, right? The blasts are building. That's how I think about it. The fibroblasts, in their nucleus, there is the genetic code that is needed to create free pro collagen. Now, when you hear the term free pro collagen, where are we in terms of organelles?
Rough endoplasmic reticulum.
RER, rough endoplasmic reticulum. Awesome job, Amina. Remember, in that RER, there's a lot of post-transcriptional or translational modification.
Transcriptional.
Did I hear transcriptional?
Yeah.
It is post-translation, right? You've translated the amino acid sequence, but it gets modified here when, since you've already done the translation, you're doing more hydroxylation, you're adding sugars, glycosylation, etc. So it's post-translational. Just be aware that it's translational, okay? Now, this is a
Can you
key.
repeat the beginning? Because I got lost.
Okay, no worries. Remember, when we're dealing with collagen, we're starting off the bat with having genetic that is pretty much synonymous across every cell. But in order to create the unique types of collagen that you're going to have, it has a lot of post-translational modification. But step by step, what we're going to do is the nucleus is going to have, the transcript being made. From there, it then goes to the ribosomes, and in the ribosomes, you get your amino acid sequence being created, and finally, you get the pre-pro collagen, and that pre-pro collagen is shot into the RER. In the RER, you have several key steps that's happening. Okay, first, you're having pre-pro collagen made. 2 is pre-pro collagen, okay? Only time you're gonna have pre-pro collagen is in the RER, okay? After that, you're gonna have a lot of modification there, and then you're finally gonna get to pro collagen. We'll cover that in a minute. First is hydroxylation. Now, hydroxylation is very important. Where are you adding your hydroxyl groups to? Which amino acid?
Proline and lysine.
Proline and lysine. Remember those 2 amino acids. It's very important. Specifically, remember proline, okay? Hydroxylation happens there more so than in lysine, just be aware. Now, which position is in what position is glycine located? That's a very important thing.
Third position, right?
Third position, exactly. Third position. That is very important. We were even asked that simple of a question in one of our quizzes and potentially in the exam. Okay. Then after you hydroxylated it, what do you do after hydroxylation?
The glycosylation.
Glycosylation. And that's what you guys are seeing here in 3. You're seeing the addition of sugar molecules, and those are presented in these cyclic structures, okay? Then, now, before we can get too far down that, hydroxylation is very important, right? Now, what is a common vignette that they give you guys for scurvy?
The vitamin C deficiency, right?
Yep, and it's typically someone who's like a sailor or someone who's out at sea for long periods of time. They haven't, had a orange in like 9 months. They have like lesions and bleeding from the gum, because that's the locations of a lot of different collagen being synthesized that are making sure that any sort of trauma that you get from eating, any sort of cuts and bruises that you've had previously, they don't reopen, is all dealing with collagen, okay? Now, that's important. Remember scurvy, that's dealing with hydroxylation, not glycosylation, hydroxylation. think of the seamen out in the ocean. They're out there. They haven't had. They don't have access to like fresh water. There's like hydroxylation. That's how I remembered it. Then osteogenesis imperfecta. Okay. Now, what is the genetic issue behind osteogenesis imperfecta? What is the allele that's affected?
A glycine is being replaced by a cysteine.
Exactly. Important. Remember that change, okay? That change reshapes the conformation of this triple helix structure, okay? Whenever you hear the word triple helix, that's 3 separate peptide sequences that have come together and created this 3D structure, okay? Three separate sequences, and that makes up the backbone of what eventually turns into your procollagen, okay? When you see this, this is procollagen, okay? Then you get further into the Golgi apparatus, right? In the Golgi apparatus, it gets further modified, okay? That's, remember, here it's also procollagen. And when do you finally see propocollagen, guys?
When it goes outside of the Golgi to the extracellular. Yep.
Exactly. Propocollagen, only time you see it is outside of the cell, okay? Outside of the cell. Remember that very, very well. Extracellular space. Now, important concept, right? It is at this point, it gets cleaved at the terminal ends, the C and the N terminus. What do I mean by C and N terminus?
The amino end and the carboxyl end.
Exactly. Remember that, because that is once again more modifications that happen, right? Those cleaving have to occur so that you can have bundles of collagen attached to each other so that they can form this nice, strong structure that you're going to eventually see here and here, right? This is the whole bundle that you're seeing here. Now, in this location, you can have several different pathologies. Ehlers-Danlos deals with what type of pathology?
Type 3 or 5.
What was that?
Type 3 and 5.
Type 3 and 5. Exactly. And we're gonna go over those individually. And then Menkes disease is very unique. What is Menkes or Menkes disease dealing with? Copper deficiency. I see you guys saying it in the chat. Copper deficiency, very important, right? Copper deficiency here. What is the deficiency here? Vitamin C, okay? Don't switch up those two. This is happening inside the cell. This is happening outside the cell. I know I'm spending a lot of time on this particular slide, but this is one of the very, very tough concepts that can be tested, and they can kind of twist the vignettes in order to ask you guys the exact same thing, but in a more clinical way, okay?
Can you please repeat the cleavage thing that you said, the cleaving, the nitrogen and
yes. So there is the C and the N terminus, so which is the ends of these triple helixes, right? They get cleaved off. That cleaving is very important to stabilize it very well. If that stabilization is affected in any way, you're not going to get nice, clear bundles that you're seeing here. Because remember, the individual tropocollagen, this is called tropocollagen, they have to bind to each other, right? That actual binding deals with it's called cross-linking, right? Cross-linking is done with lysyl oxidase, okay? Lysyl oxidase is super critical, okay? And the cofactor for that is copper, right? That's what I asked you guys with Menkes disease. It's the copper deficiency that doesn't allow for this nice cross-linking at this site, AKA Menkes disease due to a copper deficiency.
I had a question, sorry. For making tropocollagen, do we need to know the enzyme that cleaves the procollagen into tropocollagen?
I believe you don't need to. I didn't see a question on it. They only like to really dive deep into each of these highlighted boxes where you're seeing like scurvies, osteogenesis imperfecta, that's with the whole genetic component, And then here they're seeing like more of the cleaving, right? So cleaving, and then genetics. Good?
Good.
Awesome. Let's go to the next one. Now, extracellular cross-linking strengthens the collagen fibers. What's the function of lysyl oxidase? It is basically deaminates the lysine and the hydroxylysine residues. Basically, it removes an amine group. The cofactor, like I said before, copper, aldehyde, and forms aldehydes known as allylysines or hydroxyallylysines, okay, residues. And that allows them to kind of basically have these connection points for which you can have covalent bonds with other structures and eventually form nice thick collagen bundles. Let's do this question and see how you guys are doing. I'm going to give you guys a little bit shorter amount of time so that you guys, so we can finish somewhat on time and you guys don't. Okay. Let's see how you guys did. Yes. So the answer is D, right? And what is the diagnosis? We said it's scurvy. What is the deficiency in, guys?
Vitamin C deficiency.
Vitamin C deficiency. Awesome. Exactly. And what's the enzyme that's affected?
Prolyl hydroxylase and lysyl hydroxylase.
Awesome. Exactly. Those are the two to keep in mind that requires vitamin C. Awesome. And here's how it looks. It's decreased stability and the tensile strength of collagen, so it can quickly degrade. So you're going to see bleeding gums, hemorrhages, and poor wound healing. And you're going to see wounds that have previously been like that you've sustained and healed over, they'll start to reopen, which is kind of scary. So just be aware of that. Any kind of buzzwordiness with these will lead you to the fact that it's a vitamin C deficiency therapy. Okay. Stabilization, it is a triple helix. This is one of those stronger conformational structures. So hydrogen bonds between the 3 alpha chains. Remember, it's an alpha chain. Don't say anything, anything else. It's always an alpha chain. The bonds that happen are between glycine, proline, and hydroxyproline. And the enzyme for these in terms of hydroxylation and glycosylation are important. Okay. Hydrogen bonds are non-covalent, and it's important to involve these bonds in the formation of the hydroxyproline residues. Okay, let's go over osteogenesis imperfecta. Now, is this osteogenesis imperfecta vitamin deficiency or is it an inherited disorder?
Isn't it inherited?
It is inherited, right? So this is inherently the problem in this case, rather than it being like, oh, I stopped having like oranges for like 10 months, okay? So just be aware of that, it's an inherited. And what is the inheritance pattern for OI?
Autosomal dominant.
AD, I see it in the chat too. Good job, guys. Make sure you really touch up on the FTM1, list of dominant, recessive, etc. Okay. It's also called brittle bone disease. 90% of the patients, it's an autosomal dominant inheritance. COL1A1 and COL1A2. Be aware of those genes, okay? Those are very buzzwordy. They're not going to tell you sometimes osteogenesis imperfecta. What they will say is COL1A1, pathology, disorder, etc. Now, which one is the mild form? Is type 1. What is type 2? It's the more severe form, but what is it also called?
Is that the congenita?
Genotype? Yes, exactly. Now, here we're going to see a couple of other kinds. You're going to see type 3 and type 4. Those aren't as much tested because the most common form is type 1. It's the mildest form, and it's quick and easy to kind of identify and diagnose. Okay? Whereas for type 3 and 4, you're going to see one girl with like exhibit A. You're going to see like dentino imperfecta in type B. You're going to, well, I mean, in type 4, you're going to see in picture B, you're going to see look at like the amount of space in between her vertebral columns. Okay, you're going to cover more of these vertebral structures in MSK, but you can see they're giving them bisphosphate injections to kind of counteract it. You don't need to know the bisphosphate part. What you do need to know is that type 1 and type 2 are very severe. That's what Dr. Trotz emphasized a lot for us. Now, Ehlers-Danlos, pathology dealing with genetics, or is it a deficiency? Like a diet or that type of deficiency? Genetics, exactly. Now, it's a mutation of type 3 collagen. What type of collagen is type 3, guys? I mentioned it before.
Utricular.
Utricular, exactly. Retricular, I like to think it's like with an accent. And then type 4?
Basement membrane.
Basement membrane. So you're going to see you have to make sure you tie those things in. It'll help it stick better. It's a large group of connective disorders. The most common thing is that it's going to have extremely stretchy skin, and it's like allows for like people to do kind of like these crazy like bendy tricks that is just wow. But in type 3 collagen disorder, it's leads to fragile skin and vascular vessel walls. It's the most severe form as it's associated with potential arterial ruptures. They call it aortic dissections, and you're going to see a lot of those. Okay. Now, type 5 is pretty wild. It's called the classic form, and it's most caused by a defect in type 4 collagen, right? The patient shows with the hypermobile joints. Be aware of that. Type 3, bad. Type 4 is a-okay. You're going to be just fine. Okay. Let's do this one. You guys are doing so well. Some of these concepts are tough, and you really have to sit with them and brush up on FTM1 stuff. But you guys are doing great. You guys are tying it all together. Lanky is not a technical term, that much I can say. I'm glad we can help, guys. This material is tough. FTM is tough in its own kind of way. So you guys are doing swell. Okay, this is correct. So very tall, very skinny type of person with sudden changes in vision, lens subluxation, tall, thin for his age, died from a ruptured aortic aneurysm, physical examination, long, slender fingers, ligaments, laxicity, abnormal ratio. Okay, all of those things are pointing you to fibrillin, right? And fibrillin is dealing with what pathology? As wow, I don't even need to say the questions. Marfan's, exactly. And that's what we're looking at here. Marfan's, right? Now, elastin, or sorry, yeah, fibrillin is very critical, right? It's a very elastic fiber. It allows for the your aorta to take in a lot of pressure and doesn't allow for it to, basically rupture or dissect in any way. And it's an autosomal dominant disorder. Remember that, dominant, Marfan's, and it's a fibrillin 1 pathology. Okay, so fibrillin, it is commonly associated with Marfan. So just tie those two words together: tall stature, long limbs, fingers and toes. What is the long limbs and toes called medically?
Arachnodactyly, right?
Exactly. Coming from the Latin roots of like spider, like dactyle, which is like, you know, like arachnodactyly in fingers, essentially, digits. Cardiovascular manifestations, including aortic aneurysms, they call it aortic dissections or aneurysms, and then mitral valve prolapse, because your mighty valve, mitral valve, takes in a lot of pressure. So you're going to cover more about this in CPR. Okay. Now, eye-cell disease, okay, lysosomal storage diseases, they come in all shapes and form. We're even coming up on eye-cell disease again in term 2. So just remember these things. You've got to remember for the long run. So in eye-cell, what does the I stand for in eye-cell?
Inclusion.
Inclusion. And what is the specific disorder in eye-cell? What can the lysosomes not take, right? Let me rephrase that. What specific thing can't be added to the, there you go, mannose-6-phosphate. That's what I was looking for. Good job, guys. It cannot be degraded. It's basically, it's a problem. Where is mannose-6-phosphate?
It's in the Golgi, right?
The Golgi.
Awesome. Golgi apparatus adds the mannose-6-phosphate tag, which then tells it that it needs to be designated for the lysosomal storage or / degradation. Okay. Common things, they're going to have seizures and other issues, early mortality. So just be aware of that. Now, here's a summary of all of the different process of creating collagen. I put this in there because it kind of pulls it all together, where each the pathology is, where you're going to see pre-pro collagen, where you're going to see pro collagen. Just be aware of that, and it'll help it keep it straight. This is pulled directly from First Aid. That being said, we are now going to transition into pharmacology domain. Sarah, take it away.
Okay. Before we start the farm stuff, so I know that when we were learning the pharmacology stuff, everyone was really stressed out because we all came across practice questions that seemed a little difficult to do, but our exam questions were very straightforward. Like if you understand the concepts, you know how to mess with the formulas enough to use them in different ways, then you'll be fine. Like just try to put this whole thing in your head as a process instead of learning it section by section. Try to like follow a drug from ingestion to excretion or clearance, and it'll kind of make it easier to follow. So let's go to the next slide, please. Okay, so I know that there's like a million slides on different ways of administering drugs. Pretty much the point of it is, you know that if you have IV infusion, you're getting all of that drug into the blood because it's going directly into the blood. All of these other forms of administration are going to have less of a bioavailability because you either have the liver that processes it and you lose some of that drug through the first pass effect, or you're not getting it all into the blood in a timely manner. Like there's a bunch of different ways that it can decrease the potency of the drug or the efficacy, but pretty much you just want to compare everything to 100%, which is your IV infusion. Let's go to the next slide. Okay, so you guys looked at absorption, distribution, metabolism, and excretion, and you guys spent a lot of time on each one of those. So we just kind of put together some of the slides that we thought were pretty important, and then the rest are just practice questions. And I would recommend doing every single practice question that is posted on Sakai for you guys, and we will always be happy to like answer your questions or any confusion. So you guys can just like message us if there's anything confusing. But if you guys can do those questions, you should be fine. Okay, bioavailability, again, it depends on the route of administration, and you're pretty much trying to calculate how much of this drug that I just ingested is actually making it to my blood, so that you can kind of figure out how much of the dosage you would need in somebody, and then how much is actually being processed and used in your body and not just being excreted. So with this bioavailability graph, you're looking at the area underneath the curve. So this blue curve over here is your control, or your IV infusion. So you're comparing everything to this because this is showing you 100% bioavailability. And then this orange curve underneath it is just comparing it to oral ingestion of a drug. So like if you pop a pill or if it's being injected through an IV, this is how you're going to compare how much is actually being distributed into your blood. Yeah, you just have to know this formula and be able to use it. Next. Distribution. Distribution is going to have a lot of different factors that come into play. One of the main ones that you guys want to keep in mind and understand is the blood-brain barrier. So with the blood-brain barrier, you have all of these structures that their sole function is to keep things out of the brain. So you're trying to block off that barrier and make it very impermeable to the things you don't want in the brain. So you guys should know that the endothelial cells are going to have tight junctions so that things can't pass between those, or paracellularly. And then you have your astrocytic end feet, and your astrocytic end feet are pretty much just like an additional covering that is going to keep things out. The P glycoprotein, I know that was one of the slides in your lecture. Just understand what the P glycoprotein is going to do. So in this case, it's going to actively transport drugs into the systemic circulation, or pretty much like out of the brain. And then to enter the CNS, one of the things you want to keep in mind is you want things that are highly liposoluble, and or they have their own active transporter that will shuttle them into the brain.
I actually have a question.
Yeah,
so the P glycoprotein for the brain, it impede drugs from getting inside?
They shuttle drugs like actively. So it's pretty much saying like, if you want this thing to move, you're gonna have to use energy for it. So when you're talking about your P glycoprotein, it's like these are things that really do need to be moved into the systemic circulation. And then it's not just the brain that uses the P glycoprotein, I believe.
I know. And the other, sorry, go ahead.
No, go ahead.
And that was my question, because I thought, sorry, I'm tired. And in the brain, it's impeding drug from getting inside the brain, or is it like just taking what the brains cannot take and just put it back into circulation?
So it depends. In the brain specifically, I believe that it's transporting drugs into the systemic circulation, so away from the brain.
Okay.
But someone correct me if I'm wrong, because it's been a long time since we've done this.
No. You guys are both saying the exact same thing. Remember, you have endothelial cells that are lining the outer border of your blood-brain barrier, right? These cells, like every cell, can allow for drugs to pass through, right? Especially if they're liposoluble. Things will get in. They will always get in, even if you have tight junctions and all sorts of systems against it. That's why you're especially these particular endothelial cells, they have the P glycoprotein, which is going to actively pump out anything that comes in back into the lumen so that it can go back into circulation. Basically, you're decreasing the likelihood that it can permeate further into the basal lamina and eventually into the central nervous system.
Yeah.
The way
So when you're talking, sorry, go ahead.
The way I understand pre-glycoprotein, they are always try to do efflux.
Yeah, that's how I understood it too, because one of the examples I believe that you guys got in lecture was the P glycoprotein in the intestines or somewhere in the digestive system. And so that one is responsible for moving the drug out of the cell and into the lumen or the blood. So pretty much you're trying to just move things out. And if you think about it, you don't want an accumulation of drugs. Like let's say you have this drug that's in that has crossed your blood-brain barrier and it's done its job, you need to get that drug out, because if you just keep adding more drug into that place, you're concentrating it in one area, and then so you can end up with like toxic levels of the drug, or like pretty much you're going to keep it there after you've already used it. So you just want to like move it out so that it can go on to the excretion part. I hope that makes sense. Okay.
Thank you.
No problem. So for this next part, with your CYP, can't speak, with your cytochrome P450 enzymes, you want to just differentiate between inducers and inhibitors and see what they're going to do. So I know that you guys got some drugs. Make sure that you memorize which category these belong to and then how they would work. I believe they gave you guys 2 examples in lecture that you should be very familiar with and just be able to use these other drugs in that place also. So with your P450 inducers, the example that we got here is rifampin and warfarin. So let's say you take rifampin and that is going to increase the metabolism of warfarin. So when you increase the metabolism of warfarin, then you're pretty much saying like, it's gonna get into the body, it's gonna get metabolized super quick, and then it's going to get excreted. With your inhibitors, you have a bunch of different ones. The grapefruit juice is the one that we kind of went over the most, and just to be very familiar with it. So your grapefruit juice is going to increase the warfarin in the blood. So it's going to decrease the metabolism of that drug. And so that drug is kind of hanging out for a long time, but it could not get to those therapeutic levels. And I think the next slide kind of shows it in the graphs. Sorry, I totally confused that in my head. With the enzyme inhibition, so this is the grapefruit juice one. This is the one that if you have it in your system for a very long time and it's being metabolized very slowly, then your whole process is just slowed down. So by the time this part gets metabolized, you already have more coming in because you're assuming that you're metabolizing things like everybody else. So if you take a drug that has an enzyme inhibition by the CYP450s, then that drug can reach toxic concentrations because it's just remaining in your system for a very long time. With the induction ones, like St. John's wort, that is going to increase the metabolism. And so you are getting the drug, metabolizing it, using it, and it's getting excreted. So you could not ever reach that level where you see that therapeutic like consequence of taking that drug.
I'm sorry. You did amazing, but like I didn't get it. I'm struggling in this area. I'm really sorry.
No, it's totally fine. This kind of took me a while to really understand, but pretty much like these are enzymes that are either speeding up or slowing down the metabolism of certain drugs. So when I take something that I am, let's say I have enzyme inhibition because I'm taking grapefruit juice with a drug, then that drug is just going to be metabolized very slowly. So I am taking 1 dose right now, it's being metabolized. I'm assuming that I'm metabolizing like everybody else. So by the time my next dose comes around, I think I've excreted and used up that drug, but it's still in my system. So I'm adding on top of that more drug. So it's kind of like saying that you're assuming that you're moving at the speed of everyone else's metabolism, but yours is slower. So you just end up with more drug in your system than you should. And so you can very easily reach toxic conditions where you've overdosed on a drug or taken too much of a drug because you don't know that your body is just metabolizing that drug at a slower pace. Does that make sense?
Thank you. I appreciate it.
Okay. If you have any more questions about this, we can kind of like go through it slower at the end. But if you just understand those 2 concepts and know the drugs, that should be more than enough for the exam.
Thank you.
Move to the next slide. Okay, let's do a question.
Just want to ease some tension with the drugs. I swear to you guys, it feels like you guys are going to turn into like a Pharm D major at this point. I swear they're not going to make it that hard. It's very simple, as Sarah said. They literally, in one of the questions we were given was like, oh, this person seems to have an exacerbated effect with warfarin or like with morphine. What would be the inducer, or which would be the inhibitor, or etc.? Like, that's as simple as they asked, and then they just gave us a list of like of drugs that could either be the inducer or could be the enhancer. So just be aware of that. Don't freak out too much about this. There's like a nice first aid list of drugs. You can look at that too. We didn't put it here because we wanted to kind of stick with what they gave you in lecture and not overload you guys.
I would say more important than like understanding the processes of the cytochrome P450s is memorizing these graphs and what alters these graphs, because I think that this was emphasized a lot more on our exam than like the details of the CYP450s.
Yeah, I was just gonna say that. I want to say we maybe got like 1 or 2 questions total on that, whereas this stuff was like a lot. And then the drugs that you'll go over this coming week, a lot too, but yeah.
Yeah. Okay, so looks like most of you guys got this. When we're talking about KM, are we talking about your x-axis or your y-axis?
X-axis.
X-axis, exactly. So when we're talking about drug affinity, then we're pretty much saying like, are we moving to the left or to the right on our x-axis? So for this one, that is why B would be the best one, the best option. If we're talking about C, then we're saying that this drug is decreasing in efficacy, and the question says you don't want to compromise the drug efficacy. So that's why B would be your best answer for this. Let's move to the next one. Okay, and here we go. So if we are moving on your y-axis, you're talking about the Vmax. So that's the maximum amount of that drug that is going to be bound and acting at any given point. The Km is going to give you more of an accurate representation of the affinity that your receptors will have to that drug. Next one.
Can I ask you one
And
thing?
again, yeah, go ahead.
So if the options had the curve A and KM, so that would be the answer for the above question, because the option doesn't have curve A.
Well, curve A is the one that we're talking about, right? So drug A, when in the question it says drug A is what we are being compared to. So
Okay.
you're looking at A and trying to see which other one is going to give you the curve that you're looking for compared to curve A.
Got it.
On. Okay, so again, know these graphs. No full, no partial, no antagonist. Inverse agonist, we didn't really focus on as much, but just know what the graphs look like and know what's causing this shift. And that should be enough for the exam. You guys do have 2 graphs that look the same, I believe, in your lecture slides. Just make sure that you do all the practice questions you're assigned, because it'll really help you differentiate between every single one of these graphs. Okay, next, let's try this. Okay, perfect. So most of you guys are getting this. When we're talking about KM, are we talking about increased or decreased efficacy or affinity? My bad. Oh, never mind, it's on the slide. So we're talking about a higher affinity for a lower KM. So when we're looking at this, the KM for compound A is going to be lower than compound B because compound A has a higher affinity for your 5-HT1B. Next question.
Just to tell you guys while you guys are clicking in, it might seem like, why the heck am I relearning all of the concepts from like biochemistry? But I swear that this is like important. We're even getting tested on it till this day. We're in term 2, and we had questions on KM, on like stuff with glycolysis and everything. So it's not completely out of the blue or the relevance of medicine. So just be patient with the material, do a lot of practice questions. It's going to stick. I'm just going to give you guys all of the positive vibes while we're here.
Okay, so again, most of you guys are getting this. And it looks like you guys are familiar with these graphs, which is great. For this one, which other graph will also look like curve C, and that's your non-competitive reversible inhibitor. So just make sure that you guys are familiar with all the possibilities of these shifts in the graphs. Let's move to the next one. Okay, so first aid is actually a really good resource for these graphs. I didn't use it when I was taking FTM2 stuff, but I kind of wish I did because it's a really good summary. So it'll show you the graph and then it'll also point out what is happening to cause this shift in the curve. And that's pretty much what you want to be able to do for the exam. So just know the differences between all of these, what's causing these changes. Is it potency? Is it efficacy? Is it the KM? Is it the Vmax? And that should be great for the exam. Let's move on to the next slide. All right, let's do this.
We were a little split on this, but most of you guys are in the right track. So I will explain this one. The answer is D. Okay, let's take a quick peek at the question stem. Aspirin is a weak organic acid, pKa, pKa of 3.5, which is the following, represents the percent of the given dose of aspirin that will be in the liposoluble form in the stomach at a pH of 2.5. Okay, So what we need to first think about is absorption, right? This is kind of an absorption level topic, and of course it deals with like excretion too, but we'll get there. Important thing with this is that you need to look at drug absorption, okay? Drug absorption, most protonated form of weak acids is the more liposoluble form, aka in the stomach, right? And the unprotonated form of a weak basis is in the more liposoluble form in the intestines. Now, why does this make intuitive sense, right? If we have a charged particle, can it enter the cell? No, right? When it's ionized, when it has a charge, whether positive or negative, it can't cross the cellular membrane. So we typically need to augment it further so that it becomes liposoluble. Lipo meaning fat, and it's soluble meaning it has the ability to get into the adipocytes and other things, right? Like that's a little bit later on. But in the stomach, it's a very acidic environment. So it needs to be protonated, right? You need to be a protonated form so that it doesn't have a charge and it could cross through. Whereas the intestines, it needs to, the pH drastically changes. So now it can be in the unprotonated form in order to become more liposoluble. And this, once again, deals with acids and bases. It's a concept that we covered time and time again in undergrad, but what you need to remember is this chart. Honestly, I know I had a huge difficult time with this, but I swear to you, look at this graph, put it like on a sticky note on your like desk or anything so that you're constantly looking at it, because these numbers, they can ask you as simple as how much is in a protonated form and how much is in a deprotonated form. And all you have to do, it's a very simple calculation. It's a simple one-to-one correlation of like, okay, if pH minus pKa is equal to 1, then they're going to talk to you about whether it's in the stomach, if it's in an acidic environment, whether it's in a basic environment. And based off of that, you can say, okay, since it's in an acidic environment, what do I need it to be in, guys? Protonated or unprotonated form?
Protonated, right?
Protonated. Okay, let's take a quick look. Weak acid, protonated form. It makes sense, right? You can sit down and watch the biochemistry video on this, but it makes some intuitive sense that it needs to be in protonated form. So based off of that, how much percentage of the drug is going to be in the protonated form? 10%. Okay. You can just play around with the numbers and you'll get the concept very well. Do a lot of practice questions. I believe I don't think Dr. Dasso is giving you guys lectures this time, but they will be posted on Sakai and they will tell you the values of pH minus pKa based off of the environment, right? Whether stomach or intestines. And it can be very simple as pH minus pKa, what's the value you're getting it? -1, and then you can be like more is in protonated form, or like more is in unprotonated form, etc. Okay.
Okay, let's do another question. For the questions that have math, like you guys have to do math for them, I know that you guys have the calculator, you have the notes section. I highly recommend that if you're able to, practice doing all of your math in your head from now, because the more you practice doing it in your head, the more easily you'll be able to do that and juggle all of those numbers in your head during the exam. And then you can always use your notepad if you need it. But I know that I made it a point to never write anything down because I wouldn't be able to on the exam. And it definitely saved me a lot of time when it came to the exam. So just practice doing it in your head if possible. If not, write it down, but try not to take too much time writing things out because, again, you're kind of like training for exam day. Okay, so we are super split on this one. So when it comes to the half-life, you want to remember that 4 half-lives is going to give you your steady state. So once you get to the 4 half-lives, then you're going to assume this person is at steady state, and that is a concept that you really do need to understand. For this one, we're told that the half-life is 2 hours. So every 2 hours, you are either increasing or decreasing by half, depending on what you're asking specifically about the half-life. When it's telling you the infusion began at 7 a.m. and then at 11 a.m., that's when we took that measurement or the blood sample, then you want to calculate the time between that. So you have 4 hours between that, 4 hours, and then 2 of those are 1 half-life. So we're looking at 2 half-lives. In 2 half-lives, can someone tell me what is the percentage of drug that we're looking at? I'm sorry?
75.
75, exactly. So I have 75. I want to get to steady state, so I want to know what 100 is. So you pretty much just set it up as like cross multiplication. 6 over 75 times 100 is going to give you what 100% should be. So when you do that, you'll get 8 milligrams as your 100%. So you're pretty much solving for the total. Does that make sense?
Sorry, could you go over how you got 75?
So in 1 half-life, if we're talking about infusion, then you're getting 50% in 1 half-life, because every half-life you are either doubling or decreasing by half, whatever concentration you're looking at. So in this situation, we started out and 2 hours later, I have 50% of what I'm looking for. 4 hours later, I have 75 because 50% is the first one. 50% of 50% is the second one. So you're pretty much adding 50 and 25. Each time it's going to be 50% of whatever I just looked at. So just if you guys can't like wrap your head around the concept, then memorize that chart. I don't know if we put it, I think we did put it in here at some point. So it should be like in 2 questions. This chart pretty much like shows it to you guys, and then there's the graph that will also demonstrate it. So just be aware that your first half-life is 50, your second half-life is 75, your third is like 84, I think, 80 something, and then your fourth is close enough to 100. It's like 90 something, really high. It's close enough to 100 that you can just assume steady state has been reached, which is 100%.
So whenever we start an infusion, that's already the half-life, that's already 50%?
No, so we're saying that 2 hours later, we had 50%. So
okay, sorry.
9 a.m. I had 50%. And then from 9 a.m. to 11 a.m., I had 50% of the 50% remaining. So now I have a total of 75% after 2 half-lives. Does that make sense?
Yes, sorry, thank you.
No, no problem. Like, I understand that this is kind of confusing to talk about, especially like because I'm just saying it out loud, but do practice problems for this. It'll make a lot of sense, I promise. Let's try another
Do you
one.
have the solved calculation for the one that we are doing? The above one?
The solved calculation, let me see if I can annotate. Can we go back to that one? Thank you, Kishore. Annotate. Okay, excuse my drawing. I'm totally doing this with my trackpad, but I started out with 6 milligrams. Assume that there's a per liter there, right? So 6 milligrams is giving me 75%, and that you just kind of have to know because it's 2 half-lives, right? And then,
yeah.
Thank you for typing that in the chat. And then you're also going to multiply this by 100 because I want 100%. So I need to figure out what 100% of this is. And that should give you your 8 total. Does that make sense? So you're dividing your starting number with however many however much percent that starting number was. So we said it was 2 half-lives, so we know 6 milligrams per liter is giving me that 75% of steady state. And then now I'm just trying to figure out, well, what's 100? And it ends up being 8. Does that make
Thank you.
sense? Okay. Let's move on to the next question. We are almost done, guys, I promise. Okay, let's try this. Okay, so most of you guys got this one. And you're pretty much just, again, trying to figure out how much the drug is going to remain in the patient's body 6 hours after administration. So by first order kinetics, 200 milligrams, and they're telling you that 200, sorry, you're starting out with 400 milligrams, 200 of those, so 1 half-life, is going to be eliminated in the first 120 minutes. So we're given enough information to assume that 1 half-life is going to be 2 hours. And then all you have to do is try to see, okay, if 2 hours gave me 50%, then 4 hours should give me 75%, and then 6 hours should give me my steady state or 100%. And then this is what I was talking about.
Sorry, could you go over? I'm so confused by these. Like, these two are my most weakest parts, and I just don't get half-lives at all, like how to calculate them. Because I think I'm just getting confused by like how to calculate. Sorry, I'm just confused.
That's totally understandable. You guys just went over these concepts this week, so it's going to take a while to get used to it. But let's look at this graph, and then hopefully that can make a little bit more sense. Can you guys see my mouse, like my cursor moving?
Yeah. Are you the one who's moving that?
Yeah. Wait, Kishore, can you like move yours away? So can you guys see this over here?
No, we can't see your mouse. Okay.
Okay. Let me see. Can you guys see it now? Yes? No?
Yes.
Okay. If not, Kishore, just jump in and point out what I'm talking about. So when we're looking at infusion, what I was saying earlier is you're looking at either infusion or clearance. And both of these are going to be concepts that you look at half-life for. So if I start here at 0, I start infusing a patient. And let's say I get to 1 half-life, I assume that 50% is where I'm at of the drug. So 50% of it is in the body. When I get to that second half-life, 50% of the remaining 50% up here is going to get administered. So now I'm at 75%. After that, I have 25% left. So the third half-life, it's going to be 75 plus 50% of the 25 that's left. The fourth half-life is going to be 87.5 plus 50% of from that 87.5 to 100. So I get to 93.75, around 94%. And that's when we start assuming that, okay, 94% is enough for me to say I'm at steady state concentration. So it's close enough to 100. Does that concept make sense?
Yeah, so that stuff makes sense, but I'm just not sure why I'm not getting how to calculate like how much is left in the body or how much is being put into the body.
Okay, so with that, usually you just are looking for your variables, right? So I'm looking for what is the amount of time that equals a half-life in this situation. Usually they give it to you as, I started here, I checked it here, and I had this much. And then where am I going to end up after this amount of time? So with that, they have given you what one half-life equals, because they said, I started here, I checked here, and they gave you concentrations, right? So I can say, if I started with this, I have 50% of it now, then I've only used one half-life. So I can figure out the time frame that one half-life is and how much of the concentration is being either gained or lost in one half-life. And then beyond that, you are just using the question stem to figure out, okay, where am I trying to get? What are they asking me about? Because they can ask it in different ways. But if you do the practice questions and try to explain every single step to yourself, it should make it clearer. I know it's like a lot to try and like understand in one go, but the more you do it, the more you will understand, as long as you keep these numbers in mind.
So if we were to take the last question as an example.
Okay.
So you're starting off with 400, and then your first half-life, 50% of that is eliminated, which is where you get your 200 after 2 hours. So then after another 2 hours, another 50% of the 200 should be eliminated, correct? So that's 100 milligrams.
Yeah,
And
so then how many
after
half-lives?
after another 2 hours, which puts you at the 6-hour mark, it would be 50% of the 100. So that should be 50 milligrams, which is what D is. That's how you guys are thinking of this, right?
Exactly. That's
Okay.
exactly how I think about it. So you just kind of got to like keep it in the back of your mind that, okay, I know the half-life time frame and I know the concentration. Now I just need to work with these numbers to get to what they're asking about. And you did it exactly how I thought of it when I was doing that question. So just keep practicing with that.
Awesome. Thank you.
No problem. I hope that made sense. I know it's kind of difficult to like understand completely the first time. All right, let's move on. So this table, I'm not sure if we really got asked about anything besides the CYP inductions and inhibitions, but just keep them in the back of your head. Obesity is going to increase the effect of half-life, the effect of obesity is going to increase the half-life, and you're just pretty much like, keep these in the back of your head. They're not going to ask you anything beyond just like recognizing what one of these are. So it's just memorizing that table. Let's move to the next one. And then also just understanding what the difference between zero order elimination and first order elimination are. So with your zero order elimination, we don't care about the concentration of the drug. Everything is going to be linear. So it's decreasing in a linear fashion, increasing in a linear fashion, pretty much saying like, I started at one spot, doesn't matter where I'm at in the process, I'm still eliminating that half-life each time. When it comes to your first order elimination, the elimination part is dependent on what the concentration is at that time. So instead of a linear graph, you'll have more of like a hyperbolic curve. And that's because I'm starting out at like 4. And then once I'm at 2, I'm not going to still be eliminating the same amount of drug that I was at 4. I'm eliminating a proportion based on 2. At 1, same idea. So I would say like first order elimination is very comparable to what we were just looking at when we were talking about half-life. Zero order elimination is just very linear, and just looking at these graphs and understanding the concepts was enough to understand this part.
I have a question.
Yes.
First order elimination, we dealing with a fraction. Is that right? Or
Yeah, a fraction of a concentration. So like instead of I'm eliminating the same amount every time, I'm eliminating this fraction of the concentration that I'm working with. The next time I'm eliminating this fraction of the new concentration that I'm working with. So yeah.
and if we get first order elimination could be same graph as zero order if they put it in log. Is that right?
If they log it, yeah. So a lot of the graphs, they'll say like, oh, if we take the logarithm of this graph, then it becomes linear. Just kind of be able to recognize that these are the same graph, but I just took the log of one of the concentrations instead of plotting the actual points. But they didn't really ask anything too beyond like that, like to understand anything really beyond that.
Thank you.
No problem. Okay, let's try another question. Okay, so for this one, you guys are pretty much getting it. Just be able to use all of these formulas and kind of like understand when I would use this formula as opposed to something else. For this one, they're talking about your loading dose. So the loading dose formula is right here. You have your VD, they gave it to you. The total concentration is going to be your 2 liters, sorry, total clearance, I believe. And they are asking you what's the loading dose. So with your bioavailability, it's 1 because they told you this was an IV infusion. So most of your questions are probably going to give you that. If not, then they'll have to give you what that value is because you aren't supposed to really like memorize any bioavailabilities for anything. So just assume that if I'm given an IV infusion, it's 1, so you can completely ignore that that variable. If not, then they have to give you what that value is. There was a question in the chat that I kind of want to talk about. So you guys do have a half-life formula. So again, like I said, you have to be able to use these formulas to your advantage. If I'm given a vignette that only gives me a VD and a clearance, then I can't really calculate how much time there is from one dose to another, how much a half-life is, because they didn't give me enough information in the STEM. So you have to be able to also calculate half-life using the formula that you were given, but the more likely questions that they give with half-life are them giving you kind of questions like we just did. So just be aware that there is a half-life formula, but it just depends on what variables you're given in the question. Okay, let's move on to the next one. I believe this is our last question. Okay, so this question was all over the place, and that's very expected. So this is like a third order question, kind of. What you're assuming here is they told you chronic renal disease. So are we talking about, like, what concept are we talking about when we talk about kidneys and renal function? What is the thing that comes to mind?
The half-life of the drugs?
Reduced excretion, and meaning that the drug will stay more in the blood.
Yeah.
Okay, so when we're talking about kidneys, the whole point of putting chronic renal disease in this question was to point out that we're talking about clearance. So pretty much what you're looking for is which of these formulas is applicable to clearance. So clearance is only a factor in one of the formulas, loading dose or maintenance dose. And who can tell me the answer of that one?
Maintenance dose,
Maintenance
which
dose.
is equal to the
Exactly.
dose and how many time you giving the dose, the dosage and how many time you're giving.
Exactly. So the dosing rate and the interval over the bioavailability, but that's not really like what we're looking at here. We're looking at which of these has to do with clearance. So the one that you want to alter is the one that's going to be affected by chronic renal disease. And that's why you would start out with the same loading dose, but we want to lower that maintenance dose because this person already has impaired kidney function. And so they're going to be clearing that drug at a slower or a less efficient rate. And that's why we would want to lower that maintenance dose. Does that make sense?
I think this slide has a lot to do with the slide that you just said to memorize, the one from the what affect half-lives, because that's what I use in order to understand it. Because if it says renal failure, I see that it increased the half-life of the drug and the blood plasma. That means if that person has take longer to clear that drugs, that means I have to lower the maintenance because the drug will still be there by the time I give the next maintenance dose.
Exactly. So to answer the question in the chat, why would we not lower the loading dose? Because the loading dose is that initial dose that gets you to the therapeutic level. So I'm pretty much saying, like, I'm giving a higher dose that first time because I want you to get from 0 to where I'm trying to get really fast, and then I'll maintain it after that with lower doses. But because the loading dose is just getting you initially to that starting point, you don't alter that, because then this patient is, if this were an emergency, for example, this patient would be taking longer to see that desired effect from giving the medication. So you always want to start with that same loading dose, but in someone that has chronic renal disease, the compromise is going to come with the clearance. So the clearance is impaired. I want to make sure that I'm not giving this person super toxic amounts of this drug because I know that their body is clearing it at a slower rate than other patients or a somewhat healthy person would. Does that make sense?
I have a question. Like, what about if that question, the same question was about, for example, they said VD, obesity affect VD. So would we have to lower the loading dose if they say the patient was obese?
So yeah, so you would look for which of these formulas includes VD as one of the variables, and you're correct, it's the loading dose. So I would have to alter that loading dose because I know that this person is metabolizing this at a slower rate. So you want to know, like, if it's metabolism that's the issue, then I want to start out slower because I don't want them to like get a high dose, metabolize it either super slow or super quick, and then for them to see toxicity or toxic effects based on that. When we're talking about clearance, all I'm worried about is getting it out. So I'm going to start you off at the same dose as everyone else, but I'm going to give you slower or lower maintenance doses so that I don't get you to that toxic level again. Does that make sense?
Yeah, it does. Thank you.
Okay, perfect. So the
Can I ask
last
a
slide.
question?
Yes, go ahead.
Sorry. Okay, because I was thinking about liver failure here. So you would
Yeah.
think about liver failures metabolized like slower, and then would you reduce the loading dose with liver failure?
So with
Both.
liver failure, it's the same idea as your renal failure. Sorry, Kishore, can we go back up to slide 107? I think that says. Yeah, right there. So this is the table that we're basing this off of. For the clearance, liver failure and renal failure, cardiac failure, all of these are going to increase the effect of this drug on my half-life, and pretty much like we're talking about the clearance for these. So you want to keep that in mind. It's going to be the same idea with renal failure, with liver failure, with cardiac failure. And then for the VD, if VD is what we're looking for in an obese patient or with pathologic fluid, then you want to keep that in mind for your loading dose, because VD is it's one of the variables in loading dose. Does that make sense?
Yeah. Thank you.
No problem. So it's just based on this table. Again, understand this table, but you just want to understand the trends of it. Okay, and then this is just the formulas that you guys were given. Know these formulas, know how to interchange them. Like the maintenance dose didn't tell me that clearance was a variable, but you have to also understand dosing rate has its own formula and clearance is a variable in that one. Okay, so we are done, I believe. If you guys have any questions, we'll be here for a few minutes. This will be posted. We can stop the recording.